Hi students, welcome to Year 11 Biology and Module 2, Organisation of Living Things. In this video we're going to jump back from our look at gas exchange in animals to just go back to plants and focus on a couple more of the key issues associated with plant structure and function. Our specific goal is to try and interpret a range of secondary sourced information to evaluate processes, claims and conclusions that have led scientists to develop hypotheses, theories and models about the structure and function of plants, including, but not limited to, photosynthesis. And that's a big mouthful, so let's see what we can do to address this particular area. I think one of the best ways to have a look at an exercise like this is to actually review our own note-taking. What sort of a note taker are you? Do you try to take notes but don't really know what it is that you should be writing? Do you take notes but can't really organise them as you write down almost everything? You can't really discriminate between what's important and just what's there. Do you take good notes but then they sit somewhere, they're nicely filed away or they're kept in a very nice neat book, but then you don't actually go back and review them? Or do you take organised notes and review and refine them regularly? Wherever you are on this checklist, you can always move forward. And one of the ways of moving forward is to examine different types of techniques that you can use in order to try and make these as successful as possible. So what are some of the things that we need to keep in mind when we're talking about secondary sources? So remember, the important thing about secondary sources is that we haven't gathered this information ourselves. We haven't done these experiments. We could do them, but we're looking at what other people have written, what other people have done, what other people are reporting on. So the first thing that we need to do is we need to put them through the CRAP test, or if you prefer, the Parker test. The key thing here is that we're looking to try and evaluate each of the sources based on their currency, their relevance, their authority, who wrote them, their accuracy, and their purpose. We do know that sometimes bias can creep into writing. There may be a reason why a particular position is being taken other than simply based on a review of the facts. So it's important that we go through this little checklist to make sure that we've got some things that we can look at. In this particular one, we're looking at photosynthesis. So let's have a quick look at an actual article. Let's have a look at this article and see if there's some things that we can take from it. How do we do this? How do you take an article like this and try and take notes from an article or try and summarize the information that's in the article? Well, one method that you could use is called skim and scan. Skim and scan are two different techniques when you're reading. The skim technique is about trying to run your eye down the page. Now, this is very different to the way a lot of people read. Most people read across the page, exactly the way that we were taught to read. So we read, people have long been interested in how plants obtain the nutrients they use for growth. That's reading every single word in the order that it's been written, or at least the order that it appears on the page in front of me. That's not skimming. Skimming is when I cast my eyes down the page. Your brain is incredibly good at filling in the gaps. The problem is we often slow our brains down to try and read every word because we think we need to take this in. Now, I would say that if I was reading this in French, I couldn't do this very effectively because my knowledge of French is not good. And so therefore my brain wouldn't know how to fill some of these gaps in. But because A, I'm reading this in English, and B, I'm reading this in an area that I have some knowledge of, if I cast my eyes down the page, I can get a very, very quick overview of what's involved in this particular article. The second thing that I can do is I can then scan. I know that when I'm scanning, as opposed to skimming, I'm now looking for some key words or phrases. Now, the key words and phrases will vary from time to time, but one of the things that's very important in thinking about photosynthesis, obviously the most important thing is the word photosynthesis, so I can scan and look for every time there's a mention of photosynthesis. But what might be more useful for me is to actually write the equation of photosynthesis down and think about what's actually involved. So my skimming and scanning gives me a little bit of an idea to overview, to get a general idea of what's going on, and also to focus. So my skim is an overview, my scan is a focus. Now, because I know that the photosynthesis process is about water, and it's about carbon dioxide, and it's about the production of glucose, and it's about oxygen as a byproduct, and it may also involve light, like sunlight. It may even involve chloroplasts. These could be regarded as significant features of photosynthesis, and maybe in this article I might be able to pull some things out that will help me to understand the history of this. As a technique that you might use, let's have a quick look at Cornell Notes. Cornell Notes have been used for a very long time, and it involves basically dividing your page into 4 different regions. So the top is just the heading. So that will give you some indication of what you're doing. So you could put history of photosynthesis, you might just put the date. You'll give yourself some indication, so when you're coming back on your notes later on, of where this fits into the bigger scheme of ideas. Then you're going to include the main ideas, a real summary section, which is going to be much thinner than the rest. And that's this section over here. This is the second that you'll put in order to give yourself places for a summary. The majority of your note-taking is going to be in this region over here. And as you'll see in a minute, that's actually one of the first things that you need to do. And then finally, you'll be able to write a summary. And if you're someone who likes to use palm cards or some sort of simplified version of your notes, transferring the summary onto those palm cards is a good way of pulling all of this information together into something that's nice and neat and simple. So where do you start? Well, the first thing you need to start with is obviously the header. You need to make sure that you put your heading in first so your notes are organized. What do you do next? Well, the next thing you do is you take notes. Now, this means your notes don't have to be perfect. If you're one of those who tick the I just don't know what to write, so I write everything category, then you're going to have big lots of long sections of notes. That's okay for now. So just do it the way that you would normally do it. The next thing that you're going to do, though, is you're then going to look at your summary. So often with Cornell Notes, people do 2 and 3 back to front. It's not about trying to get the key ideas and then expanding them with notes. It's get the notes down first. Get all your ideas down. Get some sense of the overall picture that you're looking at or the overall concept, and then how you're going to summarize those notes. And then finally, the last section is to actually pull it all together and write a quick statement. Sometimes in biology we'll put a diagram together, something that brings all of these ideas together. So rather than go through the whole process with you, here's a quick set of notes that you might have pulled out of that history of ideas about photosynthesis. So the first idea, and the Greeks were incredibly intelligent and real deep thinkers about the natural world. They just weren't empiricists. They didn't carry out a lot of experiments. So that meant that whilst they came up with some really good ideas about a lot of different aspects of science, some of them were a little bit lacking just because they couldn't actually test these ideas. So the Greek idea was that all the nutrients were in the soil. Later on, Van Helmont's experiments showed that if a willow tree, which was what was used, was weighed, a small little sapling, and it was placed in a pot, and the amount of soil that was in the pot was also weighed, and then all that happened during the course of the experiment was that the plant was watered. So we know when we water a plant, it all falls into the soil. And at the end of the experiment, the willow was again weighed, and the soil was weighed. In this situation, the willow's weight went up considerably. The soil went down by a very small amount. So there was no direct correlation between the amount of nutrients in the soil going into the plant to create all that extra mass. It must have been something else. It must have had something to do with the water. So water is one of our first key ideas in photosynthesis. Then we had Priestley's experiments. Priestley was investigating something called phlogiston, which was basically a name given to a substance that was thought to help things burn. And an experiment which was not particularly nice on the mice, but which showed that when a candle was burnt in a container and a mouse was in the same container, then something happened that caused the mouse to die. But if the same situation was repeated and there was a mint inside of that container, the mouse continued to live. So the question was the mint doing something that was either negating the effects of whatever this phlogiston was? Was it creating something that was replacing whatever this burning candle was using? Or was something else going on? Ingenhouse read about this and realized that the mint needed light. And so light is another one of our key ideas. It's the next kind of key idea. Light's very important. And he also talked about the green parts of the plant, that you needed that, he thought, as well. So that links us into this understanding about chloroplasts, or chlorophyll, I guess, if you want to look at the specific greenness of the plant. Antoine Lavoisier talked about phlogiston, but what he said was this is not phlogiston, this is actually oxygen. So when things burn, they need oxygen. When animals are trying to live, they need oxygen. But plants seem to actually make oxygen, seem to replenish oxygen if it's lost. So this idea of phlogiston disappeared, and now we had this idea about oxygen. As we continue to go through, we look at the source of the oxygen. Ingenhouse originally came back and said, okay, I think the oxygen's actually coming from carbon dioxide. And that makes sense. There's O2 in CO2 and there's O2 here. And so maybe the oxygen is coming from the CO2 with a question mark. And of course, we know that it doesn't. We know that it comes from the water. And that was confirmed by Van Niel. So this is the kind of way that you would build this up. I won't finish this off because it's an activity that's useful for you to do, and perhaps you'll use a different type of source material. In fact, if I'm going to do good research, I'd want to use a multiple number of different source materials to make sure that I'm getting consistency in all of the information that I'm getting. Maybe I've just picked a few things out here that aren't as important or don't appear in other works. So what we're trying to do is we're trying to build our picture up of what's going on. What do we know about photosynthesis? How have we gathered some additional information about the reactants, so what we need, the raw materials, which are going to be important, as well as the products and the process of photosynthesis. So each of these could be regarded as our key ideas. Our summary then might just be a statement that tells us a little bit about what we already know. Don't forget, one of the problems with science is that we don't have all the answers. We ask good questions. And in fact, the best scientists ask the best questions. And then they seek those answers. So one of the things that we're looking at when we try and give you an opportunity to read through something like this, to make some notes and to give yourself a little bit of an overview of a process like photosynthesis, is to say, what do we currently know about photosynthesis that wasn't known previously, and how did that knowledge come to us? How can we leverage our current knowledge off all of those ideas and experiments from the scientists that have carried out all of this work in the past? Thanks for watching.